In this next video, we'll look at some reactions that amino acids can undergo. Now, some of those reactions that we're going to look at are going to be important for amino acid structure and function, and others will be just unique, interesting reactions that are being performed for research. So it's important to be able to do reactions on amino acids in order to do research, because we want to make our own peptides, or we want to see how these can react under different conditions, that sort of thing. So let's just look at a few of those, and I wouldn't commit these to memory at this time. I think it's important to just realize that amino acids can undergo reactions, and that some of the side chains or the alpha groups themselves are in fact reactive. So carboxyl groups can form amides and esters. Amine groups form Schiff bases and amides. You may remember seeing that in organic chemistry. If not, you can go back and look those up. Edmonds reagent, or phenyl isothiocyanate, reacts with the amino group of an amino acid or peptide to produce a PTH derivative. Side chains show unique reactivities, so cysteine residues can form disulfide bonds and can be easily alkylated as well, and few reactions are specific to a single kind of side chain. So here we have an example of Edmonds reagent, which reacts with the N-terminal amino acid of a peptide or protein to form a cyclic thiazolene derivative that can react in weak aqueous acid to yield a PTH amino acid. So here we have the N-terminal amino acid, which obviously proteins have to have an N to them, and one end is going to have an amine group because of the way they are polymerized, and the other end will have a carboxylic acid group. So this amine group reacts in mild alkali solution with this reagent to give this cyclic product, and then we get this PTH amino acid. Here's an example of a disulfide bond, which occurs naturally in cysteine residues of two peptide chains or protein chains. So this disulfide bond occurs from these thiol linkages here that are oxidized to form a disulfide bond. That's the sulfur-sulfur linkage, and we're going to see this one a lot. This is a naturally occurring linkage that forms in order to hold two protein chains or two pieces of a protein together into a 3D conformation. Another example is green fluorescent protein. A jellyfish native to the Northwest Pacific Ocean contain a green fluorescent protein that obviously will glow under fluorescent light, and genetic engineering allows this protein to be used as a tag, and this tag can be put into an organelle or something in order to add this chromophore that can then be viewed later. Okay, and this is accomplished with the prosthetic group, which is an oxidative product of this sequence: phenylalanine, serine, tyrosine, glycine, valine, and glutamine. Okay, and so that's after oxidation, you get this species that is a prosthetic group of this GFP. Okay. This green fluorescent protein, so that will fluoresce. The yellow fluorescent protein, okay, so expressions of combinations of three different fluorescent proteins in a mouse brain produces 10 different colorations of neurons. Okay, so then the neurons can be viewed under a microscope. And this is called a brain bow, which enables distinct cells within a brain to be viewed at one time and allows scientists to study brain cells in different ways. So I don't do this type of research. I'm not that familiar with it, but if you're interested, there's more information in your book, and you can also look it up online. So that's just some different reactions that amino acids can undergo, and some of them are natural reactions and some of them are not.